Good morning, students. Hope you'll be fine. Last time we were discussing the RNA, that is the mRNA, and we will continue that topic again. We know that there are the different types of the RNA on the basis of their functions. First one is mRNA, second one tRNA, third one rRNA, and fourth one that is hnRNA. What does these mean? What are the various functions these different types of the RNA are going to perform? We will describe them, we will study them one by one. Now today we are going to discuss this mRNA. Why is it called as mRNA? It is called as mRNA because it carries the messages from DNA. So we will study what are the messages, what for these messages are in the RNA. Now this RNA has been called as mRNA. Why this has been called as mRNA? Because it consists of genetic information. It consists of genetic information copied on it by DNA. It consists of genetic information copied on it, and this mRNA, it uses that information for the synthesis to code. It uses this what we call as information to code a protein. So this mRNA, because of its functions, that it consists of genetic information which has been copied to it by the DNA, which has been copied on it by DNA, and mRNA uses this information to code a protein. So this, what we call as this mRNA, it is found in the nucleus. It is found in the nucleus. Now, the formation of mRNA. Formation of mRNA from what we call as from one strand of DNA. From one strand of DNA is called as, this process is called as transcription. This is called transcription. Though transcription is the process of formation of mRNA from one strand of the DNA. We know that the DNA is double-stranded. It has got two strands. DNA की अगर हम बात कर लेंगे, it has got two strands. But when replication means when these two strands they break up, they become apart from each other. When the replication takes place, the two strands जो DNA के हैं, they separate from each other. They in both these, what we will see, there takes place. What we will see, the two strands they takes apart from each other. Now, so first we'll see replication takes place from one of these strands. What we will see, the process of transcription takes place, and this transcription process gives rise to the formation of mRNA. So this process, it takes place. Because of this, it contains the genetic information. Is mRNA is present within the nucleus, and from nucleus, it is shifted towards the cytoplasm. From nucleus, it is shifted to the cytoplasm, and it combines with the ribosomes to form a polypeptide chain. Now, this mRNA, it has got a template. this mRNA has got a template, or we can, in the layman's language, we can say it has got a this guide. The mRNA acts as a template. This mRNA acts as a guide, as it has genetic information, for the synthesis of the protein, for the protein synthesis, as it has got this template, this guide. अगर हम बात कर लेंगे, this guide, this mRNA, it acts as a guide, it acts as a template to as it has genetic information, and that template or that guide, and that genetic information जो इसके पास है, it guides it for the protein synthesis. So we will see the reaction जो हम sort of देखेंगे, what will happen, what in short form, in precise form, what will happen. This DNA, it undergoes a replication, then we will see से DNA से हम देखेंगे सीधा ट्रांसक्रिप्शन हो जाता है, that is transcription takes place and mRNA is formed. What we will see, mRNA is formed, and this mRNA, it gives rise to the protein synthesis. And that process is called as translation. Now this mRNA, it contains a genetic information to code a particular amino acid. In the form of, it contains, what we'll say, it contains the information, and that information is in the form of codon. So the sequence of these codons, determine the sequence of amino acids. Okay, what is the sequence of codon? That determine the sequence of amino acids in a protein chain. See, we know that DNA, what we will see, the two strands of DNA, what we will see, it has got two strands. One strand is called as template strand, and other one is called as coding strand. Now, what we will see, this template strand, what we will see, it takes part in the formation of mRNA in the 5' 3' direction. Now this part which contains this part, this is called as the cistron, or this is the template strand which contains the genetic information, and it takes part in the formation of the mRNA. Now, one term is there that is called as the cistron. What is the cistron? Cistron is the segment of DNA that contains the genetic information for the synthesis of one polypeptide chain. So what is this cistron? It is the segment of DNA that contains the genetic information for the synthesis of the one पॉलीपेप्टाइड चेन कि अब कितने सिस्ट्रॉन्स है। ऑन द बेसिस ऑफ दिस सिस्ट्रॉन्स mRNA इज ऑफ टू टाइप्स। इन शॉर्ट में हम चले जाएंगे, mRNA इज ऑफ टू टाइप्स। mRNA इट इज ऑफ टू टाइप्स। एक हम बोलते हैं मोनोसिस्ट्रॉनिक mRNA, दूसरे को हम बोलते हैं पॉलीसिस्ट्रॉनिक mRNA. As the name indicates, this monocistronic it is mainly found in eukaryotes, and this polycistrons is mainly found in prokaryotes. Now, as the name indicates, this monocistronic, अगर हम बात कर लेंगे, it is having the this genetic information to code only one type of polypeptide chain. To code only one polypeptide chain. So this has got the genetic information, this monocistronic mRNA, to code only one type of polypeptide chain, and it has got the information to code more than one type of polypeptide chain. Polypeptide chain. Again, now the concept of this mRNA concept, अगर हम बात करेंगे, the concept of mRNA, mRNA was given by Jacob and Mond in year 1920 rather. This mRNA, इसके हम characteristic features देखेंगे, it is short-lived. That means it codes one or two polypeptide chain, then it dies. दूसरी बात, it has got very high molecular weight, and that weight ranges between 5 lakh to 10 lakh. It has got very high molecular weight. तीसरी बात अगर हम बात कर लेंगे, it is found in least quantities. It is found in least quantities in RNAs, that is about 1 to 5% अगर हम बात कर लेंगे of the total RNA is mRNA. So we can say that this RNA, it is short-lived, it has got high molecular weight, and that weight it lies between 5 lakh to 10 lakh. It is found in least quantities in what we call as a RNA, that is about 1 to 5% of the total RNA is mRNA. Now, if we see the structure of this mRNA, see, we will see this RNA, it is thread-like, it is somewhat long, and it is single-stranded. It is single-stranded, और अगर ये strand single strand है, one end is 5 primer and third end is 3 primer, and the direction is in 5 primer to 3 primer. If we draw the structure, now I will draw the structure in different colors so that it becomes somewhat clear. See, I have draw the structure like this. If the direction is like this, direction is in this 5' to 3', like this. Now, what is this point? This 5, it is called as cap. CAP, cap. And this cap part contains methyl, contains methyl guanosine triphosphate, and this is simply written in MGPP. This is methyl, this G is guanosine, this is phosphate. Now, then after this, we have got this. What is this part? This is the non-coding part. This is the non-coding part, or we can say non-coding region. Again, this region जो यहाँ पर है, it is again non-coding region. Now, what is this? This part, अगर हम इसकी बात कर लेंगे, this part, this is the initiation codon, TIATION, initiation codon. That means here the process of initiating the code takes place, and what we will see, that initiation codon that forms, that starts the formation of protein chain. अब जो वो कोडन है, that is mainly AUG, or you may write it as CUG. Now, this is the initiation codon. Now, what is this red, this portion? This is the coding region. This portion, this is the coding region. It contains codes. Now, what is this? This is the termination codon. This one, termination codon. And what we will see, it consists of 3. That means it stops coding. It stops codon. It stops codons and is of 3, and is of, it stops codon and is of 3 types. कौन से? UAA, this is one. Second one is UAG, and third one is UGA. So it stops codons and is of 3 types: UAA, UAG, and UGA. Now this last one, what is this? This is called as tail. This one. This is called as tail, and this tail is called as, what it is called as? It is called as poly A. It is called as poly A tail, like this. And how many there are here? 200 to 300 adenylic, adenylic acid residues are present. So this is the structure. This is the cap. This is called as the tail. This cap contains methylguanosine triphosphate, then there is a non-coding gene, then there is the initiation codon that starts the formation of protein chain and it consists of AUG or CUG. Then there is the coding region, then there is termination codon, it stops codon and is of three types: UAA, UAG, UG. Then again there is a non-coding region, and last one there is a tail which is called as the poly A tail. Here what we'll see, 200 to 300 adenylic acid residues are present. Now अगर हम बात कर लेंगे यूकैरियोट्स की, प्योर प्रोकैरियोट्स की हम बात नहीं करेंगे. लेट इफ वी टॉक ऑफ द यूकैरियोट्स, व्हाट वी विल सी इन केस ऑफ यूकैरियोट्स, व्हाट वी विल सी, जो mRNA है इट इज नॉट डायरेक्टली फॉर्म्ड फ्रॉम DNA. व्हाट विल सी फ्रॉम DNA, व्हाट हैपन्स DNA, HNRNA, जो अभी हमने फोर्थ टाइप बताई थी, HNRNA is formed, and from this HNRNA mRNA is formed. Now this term, what is this called as? This is called as heteronuclear RNA. This is called as heteronuclear RNA. And what it is called? It is called precursor. It is called precursor. Precursor किसके लिए? It is called precursor for mRNA. Precursor for mRNA. Now this HNRNA, it represents क्या? Split gene. It represents split gene. What we will see in case of this, हाँ अगर हम इसकी बात करेंगे HNRNA के, its concept was given by Philip Shaw. And what we will see that it consists of two types of sequences. There are two types of sequences. एक को हम बोलते हैं एक्सोन्स, दूसरे को हम बोलते हैं इंट्रोन्स. Exon or intron. See, if we draw the diagram, what we will see. In order to differentiate this, I am making it somewhat of different colors. This is axon. This is intron. This is axon. Just different sequence. This is intron. Then this is axon. Then this is intron, and again this is axon. Now what happens? हाँ, this axon का काम क्या है, intron का काम क्या है? Axon क्या है? These are simply expressed sequences. They can be expressed. These are expressed sequences, and simply they contain genetic information. As they express, that means they contain genetic information. Now in case of intron की अगर हम बात करेंगे, what are these? They are intervening sequences. They are intervening, VENING. They are intervening sequences and cannot does not contain genetic information, and do not contain genetic information. So what happens? These what we call the introns, they can be removed. They can be removed. Now, the removal of introns. The removal of introns is called, इसको हम क्या बोलते हैं, RNA slicing. It is called as splicing. It is called as RNA splicing. And जो इसकी सीक्वेंस होगी वो कैसी बन जाएगी, आप देखो सीक्वेंस कैसी बन जाएगी. This is exon, this is exon, this is exon, this is exon, and this is exon. Because now, अब जो मैच्योर mRNA है, मैच्योर mRNA, it consists of only exons. इसमें सिर्फ exons पाए जाते हैं. No introns are found here. In mature mRNA, we will see only mature mRNA. It consists of only exons and no introns are found there. So this was all about this structure of this mRNA. Now tomorrow we will proceed further for different types, what we call as this different types of the RNA, that is the tRNA and rRNA. So in case of this eukaryotes, this DNA, this mRNA, it is not directly what we call as obtained या formed from the DNA. Here we will see बीच में क्या आता है, hRNA, and this is called as a heteronuclear RNA. This heteronuclear is a split gene and it consists of two types of sequence, exon and intron. Exon, they are express sequences and contain genetic information, while these introns, they do not contain genetic information and they are removed from this RNA, that is called as RNA splicing. So a matured RNA, it consists of only these exons. This was all about today. Tomorrow we will start second one, that is this rRNA or tRNA. Until then, goodbye. God bless you. Thanks.